The organelles are two types. We have our membrane-bound organelles and then our non-membrane-bound organelles. And again, we're not referring to the plasma membrane in this case. Either an organelle has like a covering to it, which is the membrane, because they're doing some very important processes and physiological reactions inside, and they need a closed environment. And so those would be the membrane-bound. And then we have the non-membrane-bound. So we're going to start off with this membrane-bound organelle, the endoplasmic reticulum. Yes, ma'am.
Are you going to need to be able to pull them?
Yes, you should be able to, if there's labeling on the test or whatnot, you should be able to point out which structure is which. Yep. So the endoplasmic reticulum, its job is to make proteins, right? And so there's different types of the endoplasmic reticulum, and we'll talk about that here in a moment, right? But the endoplasmic reticulum is just this big tubular network. And so there's a opening or a space at the center of this tubular network, which we refer to as the lumen, and we're going to see a lot of processes that are going to occur with this structure here. And so there's a structure inside the cell called a ribosome. And the ribosome, as soon as you hear that word ribosome, you should be thinking protein maker. That's what they do, right? They make proteins. That's the job of the ribosomes. So on our endoplasmic reticulum, it can come in two varieties. The endoplasmic reticulum can be covered with ribosomes, and then it looks bumpy when we're looking at it on a microscope. So we call that the rough endoplasmic reticulum. And then the other type of endoplasmic reticulum will not have any ribosomes attached to it. And so that's called the smooth endoplasmic reticulum. But consider, it's very important that you know, right, that the endoplasmic reticulum is this long tubular network that may or may not have ribosomes attached to it. And so it's a big function of our endoplasmic reticulum is going to be for protein production. So this picture that you see on your screen now, right, you can see here's the nucleus at the center, and the endoplasmic reticulum is in close proximity to that nucleus there. And so you'll see this version here, the brown one here, that's going to have lots of little bumps on it. And so those bumps are, those little bumps are the ribosomes. So this endoplasmic reticulum is the rough endoplasmic reticulum. And then a little bit further away from the nucleus, a little bit more distal, that's this set of endoplasmic reticulum, and that's devoid of ribosomes. So that's the smooth endoplasmic reticulum. We're going to talk about what the endoplasmic reticulum does here, right? But keep in mind, the big takeaway that I tell folks is the rough endoplasmic reticulum makes lots of proteins there, right? Lots of proteins, and it'll help with the formation of other structures there. And we'll talk about what the smooth does too. All right, so rough endoplasmic reticulum, the ribosomes insert onto it, and so they're going to make these proteins, and these proteins will travel, all right, through the rough endoplasmic reticulum, and they'll undergo, all right, some modifications in there. So when you see cells that have a lot of endoplasmic reticulum, you should be thinking that these cells make stuff, right? Like our beta cells in our pancreas, they make insulin. So when we look at those cells under a microscope, they have a lot of rough endoplasmic reticulum. Why? To make insulin. And that's one of our hormones there. And we'll talk about this here shortly, but one of our other internal cell organelles, the peroxisome, that's a special and membrane-bound organelle that is going to be used especially a lot, all right, when we're going to be undergoing certain types of reactions when we're dealing with pathogens. Like, for example, if you were to take in like a bacteria or whatnot, all right, the peroxisomes help with oxidizing, which is essentially killing that bacteria. And so we'll talk more about that in a little bit. So keep in mind, in our rough endoplasmic reticulum, we're dealing with the proteins here, right? And we're going to be making some other structures like lysosomes. But one thing that you have to understand is that the proteins that are made in the rough endoplasmic reticulum are not going to stay forever inside the cell. They're going to be either exported into the plasma membrane, they're going to be either if they're not going into the plasma membrane, then they're either going to wind up inside of a lysosome. That's the only exception, right, for like the digestive contents of the lysosome. And then finally, they'll be used to be exported out. So whatever those membrane bound, those rough endoplasmic reticulum organelles are making, if it's a protein, it can be either in the plasma membrane or it's going to be shot outside the cell through the process of exocytosis, which we'll talk about there. And so in order to move this stuff around inside the cell, we are going to use these special kind of bubbles, which we call vesicles. And obviously, if a vesicle is going to be used for a protein that's going to go into the plasma membrane or go outside the cell, right, we're going to move it from point A to point B. We call those transport vesicles. They're going to move those proteins inside the cell, and eventually they're going to exit out of the cell there. So the smooth endoplasmic reticulum, same type of scenario in which we're going to utilize it for several different metabolic processes in the cell. For example, in the liver, we'll use it to help with detoxifications for drugs and alcohol, right? But we'll see certain cells like hepatocytes have quite an extensive amount of smooth muscle, smooth endoplasmic reticulum there. Right? And so you can see it, the functions here. The first one there is synthesis. So it's going to make stuff, right? Quite a bit. But also it's going to make a lot of lipids, and we learned what those were before, right, in chapter 2, all right, fats. But we're going to see here in our rough or smooth endoplasmic reticulum, we're going to see lots of storage of lipids and, notice, phospholipids. And what is important about phospholipids? Sorry? Yeah, nailed it. Phospholipids are going to be utilized in plasma membranes, all right? That is going to be one of the key ingredients there. So you can see the smooth endoplasmic reticulum stores it. So in case there's damage to, all right, the plasma membrane, no problem. We got these nice phospholipids that we're storing. We'll just insert them, fix up the plasma membrane, okay? And so again, big part of smooth endoplasmic reticulum, especially in the liver, is detoxification of drugs and alcohol there. And one of the big things about our liver is it does a lot of processes. As you move through this course, hopefully you'll become more appreciative of what your liver does. Right? But one of the big things is it undergoes or it creates stored sugar, right? Or stored glucose, which is called glycogen. Right? So the process of making glycogen, glycogenesis, right? The liver is responsible for that in the fed state. So after you eat something, you're going to store those carbohydrates as glycogen stored in your liver. Because guess what? When you go to bed tonight, sometime in the night, depending on what time you go to bed, but usually within a couple of hours of sleeping, your blood sugar level starts to decrease. Okay, because your cells in your body still need sugar. And so they're going to draw it from your blood. But guess what? What happens when it becomes depleted? Now you become hypoglycemic. Not a problem. The liver has these receptors that monitor your blood content of glucose. And when it gets too low, then the liver will start to break down its glycogen storage and release that stored glycogen as glucose into your blood. So you can maintain the proper levels of, remember, homeostasis, right? Homeostasis. We want to keep our blood sugar levels between 70 to 110 deciliters, all right, or milligrams per deciliter. And so we want to make sure that we keep within that range. And when we start to fall below that, no problem. The liver starts to break down the glycogen and it starts to release that glycogen into your blood to maintain that homeostasis there. And that's thanks to the smooth endoplasmic reticulum, okay, that makes that glycogen. All right, so the next membrane-bound organelle is one of my favorites, the Golgi apparatus. And they nickname it the warehouse of the cell. But what you want to know about the Golgi apparatus is this is a place that those proteins that I just was talking to you about in the rough endoplasmic reticulum, they just get made. Remember, I told you those proteins are going to go to one of three places, either inside a lysosome, or they're going to go into the plasma membrane, or they're going to get shot outside the cell. All right, so these proteins are going to go through the Golgi apparatus because maybe they need to be modified. Right? They need to be altered a little bit. And so that's what we're going to see. We're going to see these proteins go into the Golgi apparatus. They get modified. We might add some things to it, might take some stuff away, and then we're going to pack it up, and then we're going to ship it off somewhere. Okay? It's almost like an Amazon warehouse. All right? So they're going to just send it out to you. Right? And so, like I said, it could be one of those three places. So if they're going to go into the plasma membrane, or they're going to go and leave the cell, we're going to put them into our secretory vesicles. Otherwise, we're going to just most likely stick it into a lysosome, and it'll become some of the digestive enzymes inside that lysosome. So when we talk about the Golgi apparatus, I'm going to come back to this slide, I promise you. This is what it looks like. And so you can see it looks like all these just elongated sacs that are just stacked up on top of one another. And the one sac that's usually closest to the nucleus and the rough endoplasmic or smooth endoplasmic reticulum, that's usually the longest and the widest. And then when we get up towards the top here or further away from the nucleus, they become smaller. So the part that's closest to the nucleus, we call that the cis face. And the one that's furthest away is the trans face. So if you look here at our diagram here, you'll see something's coming out of the rough endoplasmic reticulum. So our rough endoplasmic reticulum made a protein or some proteins, packed it up into the transport vesicle. That transport vesicle leaves the rough endoplasmic reticulum, goes up to the Golgi apparatus on the cis face, merges with that bottom sterna there, that bottom sac, and dumps its proteins in. And then the Golgi apparatus goes to work, starts modifying, doing what it's supposed to do, depending on whatever the purpose of that protein is going to be. And it packages it up through these different layers here. And then eventually it kicks it out. Let me zoom in here a little bit. And this is what I was saying. It's got three choices of what it can happen. It can kick it out, and those contents through that secretory vesicle are going to be entered into the plasma membrane, because remember, maybe the plasma membrane was damaged, or maybe you just had a protein-rich meal, a carbohydrate-rich meal. Insulin was released in mass quantities, and so insulin now has triggered this cell to make what we call glucose transporters. Glucose is a large molecule, and we want to get it from outside the cell to the inside of the cell. So really the best way to facilitate that is to make these channels in the plasma membrane, because remember, the plasma membrane is a limiting barrier. It separates the stuff on the outside of the cell from the stuff on the inside of the cell. And stuff just can't go through the plasma membrane easily. No problem. So we make these special transporters, which are proteins, that are going to help the cell take that glucose up. That's one of the many possibilities that those proteins that are going to be put into the plasma membrane may be utilized for. Many other possibilities. The other possibility is, remember I told you, right, those secretory vesicles are either going to have proteins that are going to go into the plasma membrane, or they're going to shoot them outside the cell. And that's what this picture is showing you. We're going to get rid of those proteins because maybe those proteins could be a hormone, for example, right? Or something else that's needed, or some enzymes or whatever. Proteins have lots of different uses. And then the third option, that protein comes in from the rough endoplasmic reticulum, goes through its modifications, and then it gets stuck into a lysosome here. And the lysosome is a digestive organelle, and we'll talk about what it's going to do, but it essentially eats things, right? It digests, like your stomach digests the food that you take in. Your lysosomes are going to do something similar to that, right? They're going to digest things, right? So those are your three options when we're dealing with our Golgi apparatus there, right? And where proteins are going to wind up, right? So with the secretory vesicles there. All right, any thoughts about the Golgi apparatus? Okay. So let's talk about lysosomes then. This is a very important organelle, right? Because sometimes if you are missing lysosomes, you will have some sort of metabolic conditions that could be congenital, but also, right, you need lysosomes to make thyroid hormone. So if there's issues with lysosomes in your thyroid follicular cells, there could be a very big problem going on, right, when we are dealing with making thyroid hormone. And so some of you might be familiar with some of the scenarios and symptoms that you experience with reduced thyroid hormone production or too much. But anyways, our lysosomes, because they contain, right, digestive enzymes, we can't just have those digestive enzymes just spilling out into the inside of our cell, because then it could start to damage the inside of the cell. So it makes sense that our lysosomes are going to be, right, membrane-bound organelle, because we want to keep those digestive enzymes inside. And so essentially what will happen is your cell might take something in from outside the cell, and maybe it wants to eat it or it wants to drink it. So it'll have a lysosome come along and meet up with it and fuse with whatever it is that it just pulled in from the outside, right? In the process of taking something from outside of the cell and pulling it inside the cell is called endocytosis. We'll talk more about that later on. Right? So when we pull it in from the outside, your cell packages it up into a little bubble, and then the lysosome comes along, merges with that bubble, that vesicle, and then it'll spill its digestive contents into that, and it'll eat up or digest or break down pretty much what's ever in there. Right? So the lysosomes play a very important role. And sometimes too, when the cell decides to kill itself, it's called autolysis, when the cell just says, I'm done, right, for whatever reason. And so it will trigger the lysosomes to digest the cell. That's one form of autolysis there. Okay, so that's lysosomes. Nothing really too excited. Peroxisomes, again, another exciting type of membrane-bound organelle here, and it serves a similar type of process, but in this case, it's going to be dealing with not so much digestive enzymes, but what we call oxidative enzymes. We all love oxygen because it helps to keep us alive. But oxygen sometimes can be a destructive little molecule. And in fact, you can get what they call free, I'm sure some of you guys have heard of free radicals, maybe somewhere along the line, drink this or eat this, and it helps to decrease free radicals. And so people are like, well, what's a free radical? A free radical is essentially, the definition of it is usually one molecule that has one single electron in the valence shell and just swings around. How I describe it to people is a free radical, right, especially if it's an oxidative free radical, is imagine a little kid going into a very nice fancy store that has lots of valuable glass china pieces or whatever on the shelves, and this little kid going into the store swinging a yo-yo around. That's what a free radical will do. So that free radical will smash into things and it causes damage, much like that little kid with that yo-yo swinging it around above his head or her head, and it just smashes into things. And so it's good and bad. It's good, all right, if we're going to use that destructive free radical to kill something that's going to be harmful. Like, for example, right, you take in a bacteria, right, and that bacteria and then it gets sucked in through one of those vesicles, and that big bacteria is now isolated inside that vesicle inside the cell, but it's still living. So how are we going to kill it? We're going to merge that vesicle with this guy, with our peroxisome. And that peroxisome is going to dump the oxidative enzymes into that bacteria and punch holes in it and kill it. And so if you've heard of, I'm sure everyone here has heard of hydrogen peroxide, right? Yeah. Hydrogen peroxide is great for a good many things. Right? What does anyone, tell me, shout it out. What does anyone use hydrogen peroxide for? Wounds, yeah. You want to dye your hair, all right? You want to clean your face? You can use hydrogen peroxide because it does a great job, for example, sanitizing things. It's great, not as good as bleach. Bleach is the best, right? But these cells, these peroxisomes will make like a hydrogen peroxide type of material, and that hydrogen peroxide will kill things, right? And you have certain structures too that will also literally make bleach. Anyways, so that's the oxidative enzymes. The metabolic side of peroxisomes, and if you look up here, chemical digestion, beta oxidation, lipid synthesis and detoxification, that's that hydrogen peroxide. That's what I was talking about. But one of the things that's important, well, all of these are important here, but beta oxidation, that's the breakdown of fats. And so you start to break down the fats and make ATP, energy. Lipid synthesis, all right, so we have a way to make fats, and also we have a way of breaking them down. So it's very important. This beta oxidation process can make your cell a lot of energy, and it's great for that. Okay, the endomembrane system, again, is another membrane-bound organelle. And this is made up of, and you can see everything that's involved in the endomembrane system. The endoplasmic reticulum, Golgi apparatus, all those vesicles that I was just telling you about, those little transport and secretory vesicles, lysosomes and peroxisomes. So everything that I've just talked to you about is all part of the membrane-bound organelle system, or what we call the endomembrane system. And essentially, what it is it's just a way for us to move things inside your cell. Right? So we have to get stuff from point A to point B. Remember, those proteins, when we put them in the transport vesicle, when we dump them out into the cytoplasm, they really don't have any mechanism of movement per se. They don't have a flagellum that can move it around the cell. Right? So this endomembrane system helps to transport, right, these things that I've just talked to you about that we're making. So when the rough endoplasmic reticulum is making its proteins to go to the Golgi apparatus, well, that transport vesicle, right, is part of this system, like this train track system, that is going to pretty much move our protein from the rough endoplasmic reticulum to the Golgi apparatus, and then from the Golgi apparatus into the plasma membrane or out of the plasma membrane. So that's including all of these structures here. And that's essentially, it connects from the nuclear envelope to the plasma membrane here. I'm going to come back a couple slides here to show you what I mean by that. There you go. So pretty much what the endomembrane system means is we can get stuff from the nucleus all the way here to the plasma membrane and beyond. And so it incorporates, obviously, the endoplasmic reticulum, both smooth and rough. Could be the Golgi apparatus, but then all of these little vesicles in between. So we can get stuff from here outside the cell. Because some of you may have wondered, like, how do we get things outside the cell? Some of us don't even give it a second thought, but that's basically what we're referring to. It's that transportation system, right, between some of these organelles, the nucleus, and the plasma membrane and beyond. And so basically, we're going to be utilizing our transport vesicles and those secretory vesicles big time, right? When we want to move stuff inside the cell, endomembrane system. All right, one of the favorites, everybody knows. The nickname for this organelle is called the powerhouse of the cell. And there's a theory out there that mitochondria weren't originally native to our cells, that they were actually made by another organism, somehow migrated and got into our cells, and then became part of our normal. Again, this is just a theory. I haven't heard too much on the most recent iteration of that, but it wasn't part of our cells originally. But regardless of how you feel about it, you better be sure darn glad to have them, because they will make a lot of energy for you. Only problem is that you need oxygen to do so. Right? So normally when you break down one glucose molecule, and I know this won't really mean too much to you, but the numbers will mean something to you. So if with one glucose molecule, if I don't have any oxygen, I'll get 2 ATP, which just ATP is just a unit, it's a molecule that your cells use for energy. Okay, so it could be $2, 2 apples, whatever, but it's 2 of something. So when we break down a glucose molecule, we break down and we have no oxygen, we get 2 ATP. Not bad, not great. But if you have oxygen, that same one glucose molecule, when broken down, can give you 32 ATP molecules. That's quite a return on your investment. And all you needed was some oxygen. That's where the mitochondria come into play. They're going to undergo, all right, what we call aerobic cellular respiration. Aerobic means with oxygen. If you didn't know that, write it down and memorize that. All right, aerobic with oxygen. So in order for our cells to get that 32 ATP, oxygen needs to be present. That's like that friend that can get you into the club or the VIP bar at the club. Right? Oxygen is that friend. It gets, right, pyruvic or pyruvate, which is a molecule you don't have to worry about this semester. It gets it from the cytoplasm into the inside of the mitochondria. Without it, it can't get in. And so once it does, it'll undergo aerobic cellular respiration and it will get 32 molecules of ATP. So that's why mitochondria is a very valuable organelle, because it can help you get more energy. You just need to have oxygen. That's why in so many things that we do, whether you do yoga, whether you do swimming or whatever type of activity, they always say breathing is crucial. It's important. And that's true, because when you're breathing, you're oxidizing, right, your blood. And what I mean by that is you're getting oxygen into your blood and getting rid of carbon dioxide. All right, so this question here about membrane-bound organelles, what organelle functions in detoxification and beta oxidation? Do y'all remember? The peroxisomes. Yeah, nailed it. Right. Now, which one is involved in energy production? I just got done talking about it. Mitochondria, right on. Yep, the powerhouse. Right. So detoxification, beta oxidation is going to be the peroxisome. And then the mitochondria is going to be utilized for energy production on a large scale. All right, let's switch over and talk about the non-membrane bound organelles, the ribosomes, right? So the ribosomes are made inside of the nucleolus, which is this small structure that lives inside of the nucleus. And so they're made there, they're exported into the nucleus, and then eventually they leave the nucleus through the nuclear pores there, and they'll reside out in the cytoplasm. And so they'll either be free floating, right, which those are what we call the free ribosomes. And so they hang out in the cytosol there. Or you can have the bound ribosomes. And we saw those because those sit on the endoplasmic reticulum and they give it that rough external appearance there. And so again, those ribosomes are going to make, right, proteins that are going to wind up in one of three places. It's either going to be shot outside the cell, which is we're going to export it, or it's going to be part of the plasma membrane, or it's going to be one of those enzymes that we find inside of the lysosome. Those are the three. So that's what bound ribosomes do. The free ribosomes make all the other types of protein. So if an organelle maybe needs some protein for it, the free ribosomes will take care of that. All right. So when and again, I'm not worried about you folks knowing about the A, P, and E sites and whatnot. That's when we get into transcription and all that stuff for making protein. Just know that ribosomes, you'll have a large ribosome and a small one, okay? Okay, so you'll have what we call a large subunit and a small subunit. And what will happen is these two subunits will come together. So here's the small subunit, here's the large subunit, right? And so what will happen is between these two subunits, we'll start to add amino acids to this structure, and you're going to just start creating a protein that's going to come right out the other side. That's all you really need to know for this. So the building blocks for proteins are amino acids. So we'll start putting together these amino acids, and when you start to string enough of them together, right, at least you have to have at least over 200 amino acids strung together. I think it's 201 before you get a protein. And so that's all you're going to do. And that's what these ribosomes do. They just start sticking these amino acids onto the protein and start to make that molecule until they're done. All right, so those are the ribosomes. And here you can see on our picture of our electron microscope here on the right side of our screen, you can see, right, you have the endoplasmic reticulum with the bound ribosomes on the outside. And then you have these clusters, these little dots on the outside. Those are all on their own. Those are the free ribosomes. So when we talk about our two subunits, here's your large subunit, here's your small subunit. So they'll fit together and create our functional ribosome. And so these amino acids will start, and again, if you want to learn that process, go ahead and read chapter 3. All right, but the amino acids will be fed into one part of the ribosome, and then in another part of the ribosome, the protein will start to shoot out. Right, and so again, the amino acids come here, they get stuck together onto our protein, and they get shot out the other side. All right, the next non-membrane bound organelle is the centrosome. And so what the centrosome is you take these two cylindrical structures. Think of a toilet paper roll with all the toilet paper off. That's what these structures kind of look like, that cardboard kind of structure. They're going to be cylindrical there. And normally when the cell is not dividing, they sit perpendicular to one another. So it'll look like this. All right, so they sit perpendicular. And so normally what we'll see is in a non-dividing cell, so which is most of your cells, right? So when they're not dividing, these two what we call centrioles, and both of those together make up what's called the centriome, right? But the two centrioles, which are going to be perpendicular to one another, they're going to hang out near the nucleus, and they just do their thing. And we haven't learned about the cytoskeleton yet, but pretty much when the cell is not dividing, they're going to be helping to organize that cytoskeleton to help give structure and help with transport inside the cell. But when the cell divides, they're going to form this structure called the mitotic spindle. And the mitotic spindle is going to be almost like these finger-like projections that come out of the centrioles. So just before the cell divides, so the cell, oh my God, it's gonna be awful. Let me draw it. So just before the cell divides, what will happen is one of the centrioles will go to one side of the cell, the other centriole will go to the other side of the cell. And they shoot out what they call the mitotic spindle. We'll talk about it. These small, like finger-like projections to the center of the cell. And they essentially just take the cell and they rip it in half. So it creates what we call the cleavage furrow right down the middle. So then you have two new cells. So that's what the mitotic spindle does. It'll essentially help to rip the cell in half. And then the plasma membrane reforms on each of the two individual cells, and then you have two cells. So when we learn more about cellular division, which won't be in this class, it'll be in 211. Right, we'll revisit that concept. But for right now, just know that the centrosome actually creates that mitotic spindle so the cell can split in two. And so that's what it looks like right there. Normally, in a non-dividing cell, they'll sit next to each other perpendicular for whatever reason. And then they help, again, with running the cytoskeleton on the inside of the cell. And then when it's time to divide, they separate from each other and they go to opposite ends of the cell, and then they do their thing. All right, here's another non-membrane bound organelle, the proteasome. And its name even tells you, right? The first couple of letters should tell you what it's going to be involved in. It's going to have something to do with proteins. And so what I love about the proteasome is, right, for those of you that have ever worked in production, right, when you're making something, you want to make sure that it's working, right? Quality control. And so that's what the proteasomes do. They make sure, all right, that we're going to export proteins, so we got to make sure that they work. And if they don't, then we got to get rid of them. Your cells are very good at recycling. All right, very good at it. So if you have junk floating around in the cytosol, the cell will look at it and be like, hey, can I use this for something? I've got a partially made protein here. What did I say the building block for proteins are? Yeah, the amino acids. So the amino acids are in that partially made protein. So guess what? I can use those amino acids for something else. So this proteasome can come along and digest that partially made protein or that damaged protein for whatever. So here's what happens. Quality control situation pretty much works like this. You have a molecule that your cell uses. You ever been to a store and it has a big tag on it that says scratched or dented or damaged? And usually they'll sell like a dishwasher for like half the price, or washer and dryers. I go to the scratch and dents all the time and buy I can say $500 on a washing machine that has a scratch on the side that I'll put against the wall that no one will ever see, you know? So what happens is that your cell is going to put this molecule called ubiquitin onto damaged or unusable proteins. And it's like that sale sign. So it sticks it on there, and then a proteasome comes floating along. It sees that ubiquitin on there and it says, okay, I know that that molecule is going to be digested. I need to get rid of it. And so it will pull the protein in and then just start ripping it apart. I'll show you a nice picture here. I'll come back to this slide. There you go. So here you can see there's that ubiquitin molecule. It gets attached onto this protein, and that protein is now marked, right, for disposal. And so the proteasome comes along and it just pulls the protein down in here through the lumen, and it just starts picking apart, pulling those amino acids off. And if it doesn't get all of them, if there's a couple still stuck together, it can make peptide fragments. And then we can use that to maybe make another protein. Right? So it's just a way that we can just recycle things. And so we use it to, you know, for damaged proteins, a protein that maybe was made incorrectly, or if we don't even need that protein anymore. Right? We've made plenty, and the cell no longer needs these channels or these gates or whatever. No problem. We just pull them out of the plasma membrane and then break them down and use the amino acids for something else. And so that's what our proteasomes do. Okay, so they are going to be used for quality control, but mainly for proteins. And so they're going to go around and just chew things up. All right. So which non-membrane bound organelle functions primarily in digestion within the cell? Well, it'll be the one that we just talked about, proteasomes. All right. Specific for protein digestion, but yeah, for the non-membrane bound ones. I know organelles aren't extremely exciting to talk about. We're almost done. We're almost done. Then we'll get to the good stuff. Right. So cytoskeleton, just like we all have our own skeletons in our body, right? Your cells need a skeleton too. Right. And so our skeletons are very helpful for a number of things that when we get to chapter 7, we'll talk about that. But our cytoskeleton is going to play a huge role in one, structure and support of the cell. But look at all of these different things that the cytoskeleton plays a role in. Intracellular transport. All right, or movement of materials here. All right, cell division. We just talked about what happens with the centrioles there, or the centrosome, with the mitotic spindle. And then how we are going to place or organize the organelles inside of our bodies there. All right, but it still plays a role in which you would think a skeleton plays a role in. Helps with supporting. Right, our bones support our body. Right, the cytoskeleton supports the shape of the cell there. It plays a role in that too. So we'll see the cytoskeleton throughout the inside of the cell. And the important thing about it is too, our muscles, for example, will anchor onto our bones, right? Much like, for example, chest muscle or even your biceps muscle, it attaches onto your bones there to help facilitate movement. Well, we'll see in our cytoskeleton, we'll have these anchor points on the inside of the cell, in the plasma membrane, that we can use. I'll show you a picture here that helps to anchor certain structures to the cell, because one cell might need to be anchored to the neighboring cell. And so the cytoskeleton plays a role in that. So there's three, all right, components here that we'll talk about to our cytoskeleton: microfilaments, intermediate filaments, and microtubules. Again, not the most interesting, but something that's worth noting. So the first one is the microfilaments. Right? So think of micro being small. So these are the smallest parts. And we'll talk more about some of these proteins here in a little bit. But a good example is actin. All right, actin is used in muscle contraction. And so these microfilaments here, all right, do help to play a role in, one, obviously movement, because we're dealing with muscle contraction there. So actin plays a role in that. But also, we're going to use these microfilaments, right, when we have to undergo cell division. And we'll talk about that briefly here at the end of the chapter. But also, when we're going to create the support mechanisms for the cell, right, these microfilaments play a huge role for that. So the intermediate filaments, obviously intermediate, you should start to think, all right, we're going to go up in size. So these are going to be a little bit bigger. And so therefore, they're going to be a little bit stronger and rigid. So these will play a much larger role in the structural support overall of the cell. But I mentioned just a few moments ago how one cell might want to attach to its neighboring cell. And so if we don't have a stable interaction between those two cells, well, then you can get instability and you can start to see tissues getting damaged or start to have dysfunction. So it's nice to have the fact that, for example, in our skin, that your keratinocytes are going to be tightly packed and close to their neighboring cells, right? And that adds to stabilizing those tissues there. And so the intermediate filaments play a big role in that. And then finally, the last part of the cytoskeleton is going to be the microtubules. Now, the big thing I want you to know for these, one, they are the largest component there of the cytoskeleton, but they are also not permanent. All right, so these structures are constantly being altered, made to be longer, made to be shorter. And the reason why is, especially if you look up at the board here, when we're dealing with cell division. Once these two cells have divided, what are we going to do with all of these tubules there? We're just going to break them down. And that's what happens here, right? With the microtubules, they're not permanent. So they can be added to or they can be subtracted from, right? So they do play a pretty big role in cell division, and they play a really nice role in cell transport because, again, the microtubules are not permanent. So it's like building a road, and we got our shipment there. Right now, we're going to tear that road apart because we might need to use some of that material to make another road going somewhere else inside the cell. Right? A lot of the structures you don't want to have to be permanent in the cell because then things start to accumulate in the cell, and that becomes a problem. Things become crowded there. So that's what this picture is showing you, the different variations, right, of our different tubules here. Right? There's our microfilament, smallest at the top. Then you can see the largest one, the microtubule in the middle, and then down below the intermediate filament there, that's going to be in between these two guys. And you can see they play several different roles there. All right, now we're going to start moving into the plasma membrane, and we're going to get to some cool stuff in regards to concepts that are going to be, I cannot stress how important a lot of these concepts are going to be for you to learn, that you are going to use throughout the rest of this course and in 211 when we start talking about osmosis and diffusion and all that stuff. So I'll focus on that when we get there. But let's start off with the external surface of the cell, the plasma membrane. Right, there's three possible modifications that we can see on the external surface. And we should know all of these because we talked about them briefly in lab class last week. So the first one is the cilia, and those are those hair-like projections that we'll see. And the big thing about cilia is that cilia, the cell that has cilia on it, that cell does not move. The cilia moves, but the cell stays stationary. And so the cilia move things past the cell. So for example, we saw the goblet cell. Remember the goblet cells? Those are the cells that make mucin, mix it with water, make mucus. And so usually goblet cells were going to be found, all right, not all the time, but in most cases, they're going to be associated with ciliated epithelium. And the cilia is there to move that mucus, all right, in a specific direction, depending on where this tissue is located there. All right, so we'll see it, all right, for example, in the respiratory tract. And so that cilia is going to move that mucus not towards the lungs, but away from the lungs, right? So it's actually going to be beating that mucus up towards your oral pharynx, your throat. And some of you may have experienced this when you've had some sort of upper respiratory tract infection. Mucus production increases, and you're coughing up mucus, all right? That's because, right, one, the cilia is working hard to move it out of the way. You have some sort of issue going on, and mucus production has increased for whatever reason. And so we don't want it going towards the lungs. We want it going towards your oral pharynx or your mouth. All right, the flagella. Again, there's only one cell in the body that has that, and that's associated with movements, right? It's the sperm. The male sperm is the only cell that has flagellum, right, in humans. And so its purpose, real simple, is to move that sperm. And then finally, the last one is the microvilli. This is the one that I was telling you, right, that it's an extension of the plasma membrane, and it's a really clever design, but it's designed to just increase the surface area. Okay, because a lot of this type of tissue that has microvilli is going to be in areas that are for absorption, right? Secretion too, but absorption, for example, like in the digestive tract. So we want to increase our surface area as much as possible so we can get more absorption. So this is a nice electron microscope picture of our microvilli. So again, the plasma membrane is grown out, up, and down. And so all of this is increased surface area to the plasma membrane here of this cell. This is a really nice picture because on the electron microscope, you can see inside of the microvilli, right, some of our cytoskeleton. You can see all of these, all right, microfilaments here that are part of the cytoskeleton. So what structure of the cell's external surface is found in parts of the respiratory tract and participates in moving mucus toward the throat? Nailed it. Y'all are going to do awesome. Okay, cilia. All right, I'm going to skip this and get to this. All right, the other stuff is just talks about gap junctions and all these different types of structures. All right, but I want to get to the I say the good stuff. Let's talk about the plasma membrane here a little bit. All right, so this is one of the three components to your cell. We talked about the cytoplasm. We just got done talking about the three parts of the cytoplasm. And so now we're going to move on to the plasma membrane here. And so when we're looking at the plasma membrane, when we're looking at it strictly from the weight standpoint, we roughly have equal parts of lipids and proteins when we're discussing weight. But if you were to look at the plasma membrane, it would look like there's way more phospholipids than there are proteins, which is true. But again, the protein's way more, right? But just keep in mind when we say it's composed of equal parts of fat or lipids and proteins, it's just by the weight there. So this next statement here could not be more true. This structure regulates. What does that mean? Regulates. Yeah, it's going to be selective. It's going to pick what can enter into the cell and what can't enter into the cell. It's going to control that. And that's very important because we don't want anything just rushing into a cell. If water could just come into a cell, guess what could happen? What happens if you overfill a water balloon? It'll pop. Yeah. So we don't want stuff just coming into the cell, and we don't want as it pleases, and we don't want stuff leaving the cell, right? So the plasma membrane plays a huge role in allowing certain substances to enter and certain substances not to enter. But here's one of the things, though, and I mentioned this in chapter 2. All right, you can have two qualities will allow you to enter into the cell. One is if you're really, really small, and the other is if you are nonpolar. So that means, right, if you have both of those qualities, you can go into the cell. Doesn't necessarily mean that you will, but you can. You can go into the cell. So this is a good thing. So small, nonpolar substances. So what are we going to find in our plasma membrane? Well, obviously, we're going to have fats or lipids, and we're going to have proteins. So the three types of lipids that we'll see commonly in our plasma membrane are going to be cholesterol, obviously the popular phospholipids, and then what we call glycolipids. And that's just a lipid molecule with a carbohydrate group attached. We'll talk about some of the functions here. So just keep that in mind. All right, so let's start off with the first one, the phospholipids. These are the popular lipid that's going to make up your plasma membrane here. And we described it a little bit here when we were in chapter 2, right? But a phospholipid looks essentially like a balloon with two tails. So that phospholipid is going to have a phosphate group on it, a glycerol group, all right? And then it's going to form what we call the polar hydrophobic, I said it right. Hydrophilic head. What does that mean? What does hydrophilic mean? Yeah, water loving. So it can be around water. So you're going to find the head, right, that balloon-like structure in contact with water. And guess where what you're going to find water? You're going to find it inside the cell, right? In the cytosol, because that makes up the majority of the cytosol. And then you're going to find it outside the cell also. Right? And then the two tails that are coming off, those are non-polar. So the tails are hydrophobic, water-fearing. They don't want to be in an aqueous water environment. And so they won't be. And that's non-polar and water-fearing. And then the head, the balloon structure, is going to be hydrophilic, water-loving, and that will be polar. And so that will be in a water-like environment there. So what happens is, and normally I try to draw it, but I can't draw it very well. So when we look at our plasma membrane, we're going to see the phospholipids. They're going to line up in two parallel sheets, meaning they're going to go tail to tail. Show you what that looks like here. Y'all see on this picture here, if I zoom in at our work for me. That's the wrong one. Sorry. I'm pushing the wrong thing the whole time. All right. So we zoom in. Check it out. You see what I mean? That purple ball, that's the head. And then you have these two yellowish or orange structures coming off of that purple ball, right? That's a phospholipid. And so this is the polar hydrophilic head. And then down here, where we see those two tails coming off, that is the nonpolar hydrophobic tails. But notice we have one layer here with the heads facing out, and then adjacent to that, you've got another layer, all right, with the head facing in. Y'all see that? So it's one sheet here. No, I can't draw on this. But you have one sheet here, and then right below it, there's the other sheet. So we call that the bilayer membrane, right? It's the phospholipid bilayer membrane, right? That basically means you just have two layers of phospholipids, but they go tail to tail. So the heads are pointing out and the tails are pointing in. Can we all agree on that? Y'all understand what I'm saying? All right, the heads are facing out, the tails are facing in. So all this out here, there's water there. Well, that's okay, because the head is hydrophilic. And guess what? All of this in here inside the cell, there's water in there. But that's okay, because the heads are hydrophilic. There's no water, very little water should be in this area here. Y'all see what I'm saying? Because this is the polar hydrophobic tail area there. So there's not going to be water in that area there. All right, so that's the phospholipid layer. So we have that, the bilayer lipid structure there. And so the heads are only going to make contact with the cytosol on the inside of the cell. And then the heads on the other layer are going to make contact with the interstitial fluid, and that's on the outside of the cell. So we're just going to put the part of the molecule that likes water in contact with the water, and then the part of the molecule that doesn't like water, we're going to isolate that and put that in the middle of the plasma membrane, because it won't come in contact with the water there. All right, so that's the phospholipid. All right, the next type of lipid that you'll see in the plasma membrane is cholesterol. All right, and this is the lipid that I was telling you about that is actually good to have. Because cholesterol in your plasma membrane makes it stronger because it can withstand temperature changes if it gets really hot or really cold. And so the more cholesterol you have in there, it helps to stabilize that plasma membrane there when you have temperature changes. Like if you were to be, well, we don't have to worry about it down here, but if we're shoveling our driveway and it is, I don't know, 10 degrees out there, and then you go into your nice warm 75, 80 degree house, that's two temperature extremes. So the plasma membrane, all right, is helped to be stabilized through that cholesterol. Then we have our glycolipids. Like I said, that's just a lipid that has a carbohydrate group attached to it. and it is going to help form what we call the glycocalyx, which is this sugary coating on the outside of our cells. And again, we use this glycocalyx to help identify cells, and we'll talk more about that when you get into the immune chapter. All right, but we have cells, our white blood cells, that circulate around that are constantly inspecting everything. And so if the cell is a foreign cell, it'll have a different glycocalyx on the outside, and it's going to be labeled as a foreign or a non-self cell, so the immune cells can attack it. All right, so that glycocalyx helps us, right, for the surface of our cell to determine whether that cell is friend or foe, I guess, essentially. All right, so that's the glycolipids. So those are the lipids that you're going to see in the plasma membrane. All right, so keep in mind, you'll notice on our picture here, there's other things inside the plasma membrane, notably these big blue structures here. And we're going to talk about those. Those are going to be our proteins. So what portion of the phospholipid molecules form the internal environment of the membrane? What do we see on the inside? Right in there. Yeah, those are going to be the tails. That's going to be the nonpolar hydrophobic region. Right, so that's the nonpolar hydrophobic region inside the plasma membrane there. All right, so let's talk about the other part of our plasma membrane, the proteins there. And so obviously, if you have a protein that's in the plasma membrane, we call those membrane proteins, and there's two types. There's the integral, which span the entire width of the plasma membrane, and then you have the peripheral. And so they'll either be on the outside of the cell or on the inside of the cell. And so these proteins just float along inside the plasma membrane. Like if you've ever seen junk floating on top of the ocean, or like a boat floating on the ocean, I'm not saying that's junk, but they just kind of float within the plasma membrane. And they have tons of different functions. And we'll talk about a lot of their functions, right, in this class throughout the semester. In fact, when we get into muscle contraction and neuron action potential generation, a lot of that is reliant on proteins that sit in the plasma membrane. All right, so again, we'll go over and talk about several of these as the semester goes on. So keep in mind, the integral proteins are going to extend across the entirety of the phospholipid bilayer there. And so on some of these proteins, we'll attach a carbohydrate group. And then when we do, we call those glycoproteins. And then we have our peripheral proteins that won't span the entire width of the plasma membrane. And they're, like I said, they're usually floating on the outside of the cell or floating on the inside of the cell. And so those are the peripheral proteins. So we'll see them when we're talking about secondary messenger systems, and again, we'll get into that a little bit more throughout the course there. But just keep in mind for right now, integral proteins and peripheral proteins, those are the two types of proteins that you're going to see in the plasma membrane there. And we'll spend a fair amount of time talking about the integral proteins in this semester here. So, couple functions for the proteins. The first one that you see up there, and we'll spend a lot of time this semester talking about, are transport proteins. So when you see that term up there, transport protein, what do you think these proteins do? Yeah, they move stuff. Yeah, that's what they do. So these types of proteins are going to regulate the movements of things across the plasma membrane, whether it's something that's coming in from the outside or something that's from the inside leaving the cell. And we just talked about that, all right? When we talked about the Golgi apparatus, we're making stuff. So transport proteins, we'll talk about all of this stuff. We'll talk about some of that today. All right, the cell surface receptors. And this is something that you want to know. You want to know this term here, right? A ligand. And again, a ligand is a molecule, some sort of chemical molecule that can be moving around somewhere. And say it's a hormone, for example. So a hormone can play the role of what we call a ligand. And so hormones are made somewhere secreted by whatever made it, and then transported through our bloodstream to what we call target cells. They go to the target cell and then they cause the target cell to do something, maybe to make something. All right. So for example, we'll see, all right, when we're talking about glucose, excuse me, not glucose, insulin. Insulin is that hormone that is going to be floating in your blood, and then it's going to come to a cell and it's going to dock, like how a spaceship would dock at the International Space Station. It's going to dock with the receptor. And then it's going to send its message through that receptor to do something, to make something, maybe to stop making something. And so in this case, all right, insulin is going to tell a cell, hey, make more transport proteins so we can get more glucose inside the cell, for example. So these cell surface receptors are going to utilize these ligands, those chemical markers there, right, hormones or what have you. We'll see it in chapter 12 when we talk about neurons. All right. And so we'll see a release of neurotransmitter for whatever reason. And we'll get into all that later on. All right. Identity markers. This goes back to what I was just mentioning to you before. All right. About our maintenance of healthy cells versus unhealthy or pathogenic cells. And so these identity markers allow our white blood cells to know if a cell is what we call self, it's one of our own cells, or if it's a non-self cell, like a virus, a bacteria, or a cancerous cell. Right? So these identity markers essentially allow the white blood cell to identify the cell as being a good cell or a bad cell. If it's a bad cell, we'll initiate the immune response. If it's a good cell, the white blood cell moves on its way and goes to the next cell. All right, so these identity markers help with that. And so basically, we're just trying to determine, hey, does that cell belong to the body? Should it stay here? Or does it need to go? Do we need to kill it? Enzymes are another function of proteins. And so enzymes, what is their purpose? Their purpose is to catalyze chemical reactions. So what does that mean? Much easier to start a fire with a lighter than it is to rub two sticks together. So that's what a catalyst does. It helps to initiate a chemical reaction easier, less energy. Right? The catalyst will be that spark that initiates that chemical reaction for whatever reason. So proteins play a big role in making those enzymes. We just talked about the digestive enzymes of the lysosome, right? And those digestive enzymes will break things down so they can actually break down chemical bonds or bonds between molecules. Anchoring sites, we just got done talking about our cytoskeleton. So you'll have these anchoring sites on the inside of the plasma membrane here. These peripheral proteins, for example, will be these anchoring sites so we can secure different components of the cytoskeleton, maybe some microtubules, some intermediate filaments, whatnot. All right, if the cell is going to undergo that cell division and that mitotic spindle gets formed, we need to have these anchoring sites so we can pull the cell apart. Cell adhesion proteins, again, I told you, sometimes these cells want to actually stabilize themselves. And by doing so, they might want to create a more stabilized structural adhesion to a neighboring cell. And so these cell adhesion proteins utilize that, like a zipper. When you zip the zipper up, things are nice and tight. When you unzip the zipper, things are loose. So that's what these cell adhesion proteins will do. So all these examples here, pretty much what I was talking about. Here's an example of some of those transport proteins. You can see it's an integral protein that spans the entire width here of the plasma membrane. So stuff that's outside the cell can come inside the cell. And there's different ways of doing that here. We'll talk about all those different ways. Here you can see, right, our cell surface receptor here. Here's our ligand. That could be a hormone from another cell that then triggers this cell to do something here. Identity marker, talked about that. When it's important for our cells to recognize self versus non-self. Here's an example of an enzyme. You have your substrate. That's what you start the chemical process off with. And then that enzyme creates that metabolic effect, right? And that catalyst type of scenario here that we are able to go from substrate, what we start with, and then our finished product, what we end with, and the enzyme helps with that. And then you can see here, there's the anchoring site there, right? Various parts of the cytoskeleton merging onto or anchoring onto that protein there. And then you can see here, here's our cell adhesion protein, which is just anchoring this cell here to this other cell here, so they don't slide around. So what type of plasma membrane protein provides the means for moving materials across the plasma membrane? Transport. Yep. Transport proteins do that. Okay, so that brings us into membrane transport. How do we get stuff that's outside the cell to inside the cell and vice versa? Let's recap. The plasma membrane is a limiting barrier, right? Which means it is going to prevent things from coming in from the outside and things from that are inside from leaving the inside and going outside. So that's the purpose. I mean, for our plasma membrane, it's a physical barrier here. So I said before, it's going to regulate movements, what can come in and what can go out. And so it plays a big role in establishing what we call the electrochemical gradient. More on that when we get to chapter 12. But what it essentially will do is it will create this phenomenon or this situation in which you have more stuff outside the cell than inside the cell. For example, nature loves balance, always does, okay? And so it wants things to be equal. So if I have 100 molecules of sodium, let's say, right, nature would want me to have 50 molecules on the outside of the cell and 50 molecules on the inside of the cell, because then it's balanced, it's equal, and this is good, right? But physiology does not allow for that, right? Because there's a reason for it. So we have these pumps, literally these things, these structures right here, these pumps here in some of our cells that pump stuff from the inside of the cell to the outside of the cell, and then it'll also pump stuff that's on the outside of the cell to the inside of the cell. And so in doing so, it is going to make an unequal distribution of material both inside and outside the cell. As a consequence of that, if it's an ion, for example, like sodium, sodium's an ion, it will create more positive charge outside the cell than inside the cell. We'll go into this in much more detail. But anyways, if we have more of a positive charge outside the cell, what do you think? Does that make the outside of the cell positive or negative? Take a guess. Yeah, right. So therefore, because the outside of the cell is more positive, then by default, what's that make the inside of the cell?
Negative.
Negative. So this creates what we call an electrochemical gradient. More on that later on. All right, but I don't want to, we'll talk about way more later on in the in the semester. Okay, so this brings us to this concept here of membrane transport. How are we going to move things, get stuff inside the cell, and then take stuff that was inside the cell, eliminate it, and get it outside the cell? So there's two processes or two categories. You have passive processes and active processes. And so now we're going to talk about how these processes work and what are some of the specifics. Now you're going to learn all of these. That's a lot, I know, all right? But keep in mind, we'll go through these, right, and I'll help you through most of them, right? And a lot of them you'll just know, okay? A lot of it's just memorization. But we're going to talk about all of these processes, and we'll talk more about some than others, and that's okay, right? But by the time you're done with this chapter, you should have a pretty good idea of the different types of membrane transport. So let's talk about membrane transport. So passive versus active. Passive processes, it's even underlined up there, do not require energy, period, end of story. So no energy is utilized during passive transport. And so that's one of the qualities. Another quality of passive processes is that we're going to move a substance from an area of high concentration to an area of low concentration. Because remember, nature always wants balance, okay? So if you have a little of something somewhere and a lot of something in another place, then we want to move some of that stuff, that that high concentration of stuff from that one area to the area that doesn't have a lot, so we can balance things out. So that's what we talk about when we talk about moving substances down the concentration gradient. We're essentially going to move from one area to another area. We're going to go from an area that has a lot of something in it, and we're going to move some of that material into the area that does not have a lot. And so those two processes that we're going to talk about are called diffusion and osmosis. Active processes, it even says it in the name, so if it's active, we need energy. You think it's easier to roll a rock down a hill or push it up a hill? Yeah, roll it down. So that's how I think of passive processes. You're going to move something, all right, from an area of high concentration to low concentration. You're essentially going to roll something down the hill. Right? When we're talking about active processes, it does require energy. And now we're going to move substances up its concentration gradient. And so what does that mean? That means we're going to move a substance, all right, from an area where there's not a lot of it into an area where there is a lot of it. Right? So we're going to move it up its concentration gradient. And a lot of times we're going to be using those membrane-bound vesicles that we just learned about. Remember, we talked about transport vesicles and secretory vesicles. We're going to use some sort of bubble, some sort of membrane, right? And we're going to use it to move some of this material around. So let's start off with diffusion, right? If you look at the definition there, it's the net movement of a substance from an area of greater concentration to an area of lesser concentration. So you've got a lot of something in one spot and very little of it in another spot. And we want to balance everything out. So the area that has a lot, that substance is going to want to move into the area where there's not a lot. That's all it is. And when it moves from that area of high concentration to the area of low concentration, we say it's moving down the concentration gradient. Moving down the concentration gradient. So we're going to be referring to molecules, ions, right? And they're going to be doing the moving. And so a lot of these molecules, as you know, if you've taken chemistry, they're not stationary unless you're frozen, right? But usually when molecules move around, they're going to be generating kinetic energy. Right? If you've taken physics, there's kinetic energy, and then there's, what's the other one? Potential energy. Whew, almost forgot that, all right? Potential energy is like a rock or a boulder that's at the top of a hill. It's not in motion, it's not doing anything. But all you have to do is put a little bit of pressure on it and it'll roll down the hill. And then it goes and rolls down the hill, then it's generating kinetic energy. So kinetic energy is when you're actually having energy in motion. And so these molecules and ions are moving around. So when they're moving around, they're going to want to go, all right? Because again, balance, nature, it wants to equal things out. So when they're moving around, they're going to want to go from an area of high concentration to an area of low concentration. And once we've equaled everything out, then we've attained or reached what we call equilibrium. That means you have equal amounts of whatever that substance is. And so for the examples that I'll be using the cell, inside of the cell and outside of the cell. So we want to have equal amounts inside the cell and equal amounts outside the cell. That's what the cells are trying to achieve, but we'll talk about why that doesn't happen in some cases. All right, so when we talk about diffusion, right, we can speed it up or slow it down. So the rate of diffusion relies on two things. One, the steepness of the concentration gradient. Right? Is there a big difference between those two areas that we're talking about? So if you have, let's say, 99 molecules of sodium outside the cell and 1 molecule of sodium inside the cell, that's a pretty steep, right, concentration gradient. But if we had 46 molecules of sodium on the outside of the cell and 54 molecules on the inside of the cell, it's not that steep. There's not that much of a difference there. So the rate of diffusion won't be very fast. Right? So if there's a large difference between the two areas, right, then the steepness of the concentration gradient will be increased significantly, then there's a fast rate. If there's not much difference, then you won't have as fast a rate of diffusion. Temperature, same thing. When you add heat to an environment, it'll speed things up, right? So higher temperature, higher movement, faster rate of diffusion. That's just with anything. You add heat to what do I want to say? To an environment, right? And then it'll speed the molecules up and the molecules will move faster. Therefore, you'll see a higher rate of diffusion. All right, so that's what this picture is showing you. You'll notice when the individual is putting their medicine dropper of food coloring into the water here, initially you have a high concentration of the food coloring in this area, but not a lot out here on the periphery. But again, according to diffusion, you want what we're going to see is movement of a substance from an area of high concentration to low concentration. And that's what we're seeing here. Now, those food coloring molecules just kind of spread out, right? They gradually spread out, and then eventually it'll reach equilibrium, which means it has equal distribution all throughout this beaker. But we can see initially high concentration here, very low over here. Same thing. And so that's what we're going to see when we talk about diffusion. There's different types of diffusion. The first one is the easiest one. That's why they call it simple diffusion. Not a lot of thought processes, right? But when we're talking about the cell, because that's what this is all about, talking about the plasma membrane, right? So you have outside the cell and inside the cell. So when we deal with simple diffusion, this process, right, will allow molecules to pass right through the plasma membrane. And what two, is it up there? Oh, it's up there. All right. I was going to say, what two qualities allow for molecules to go right through the plasma membrane? It says it. Small and nonpolar. Yeah. If they're really small and they're nonpolar, which means they pretty much won't interact with anything, they can go right through that phospholipid bilayer. So they don't need help. This is good for certain things. It's great for our respiratory gases. All right, so oxygen and carbon dioxide, they don't need help getting across the plasma membrane. They can go right through the plasma membrane. But what dictates which direction they go in is determined by their concentration gradient. So for example, if there's a lot of oxygen outside the cell and very little oxygen inside the cell, where does oxygen want to go? Inside the cell, right? Makes sense. According to diffusion, it wants to go from an area of high concentration to low concentration. So oxygen, right, wants to go from outside the cell to inside the cell. What do we need oxygen for inside of our cells? We just talked about one of the membrane-bound organelles that utilizes oxygen. Yeah, the mitochondria. Mitochondria need it for cellular or aerobic cellular respiration. When do you think that we need to produce a lot of energy? Well, we're not producing a lot of energy now, right? We don't need it right now when we're sitting in class, right? When do you think that you yourself would need to generate a lot of energy? If you go running or doing what, yeah, activities, say if you got to rake the yard, mow the lawn, whatever, right? So we need to generate that energy. So don't you think we would want to be able to facilitate a process that allows oxygen to get inside the cell easier? This is it. Okay, exercising cells, like muscle cells, skeletal muscle cells, when you're working out, right, their oxygen depletion happens fast. So they become low in oxygen initially. It doesn't take long when you're exercising. So what happens is your bloodstream supplies the oxygen to the skeletal muscle cell, and that cell oxygen just shoots right through the plasma membrane and goes into the cell to the mitochondria so you can make ATP through aerobic cellular respiration. Same thing with carbon dioxide. You'll see, in fact, I don't even have to show you. I mean, talk about it. I'll show you. All right, look here. Lots of oxygen outside the cell, very little inside the cell. And you can see the arrow is telling you in which direction oxygen wants to go. Same thing with carbon dioxide. Carbon dioxide is the waste product, right, that a lot of cells will have after respiration. So you have a lot of carbon dioxide inside the cell, and very little of it outside the cell, which direction does carbon dioxide want to go? It wants to leave the cell. Where do you think carbon dioxide is going to go after it leaves the cell? Yeah, you nailed it. Whoever said that. Yep, it's going to go into the bloodstream, go to your lungs, and guess what? When you exhale, bye-bye carbon dioxide. That's where it wants to go. Oxygen, you breathe it in, goes into your lungs, then goes into your bloodstream. Then the bloodstream brings it close to these cells, and then oxygen can enter in. That's the process that we're seeing here when we're talking about, all right, diffusion. It's great, all right, for our respiratory gases. Some fatty acids, well, yeah, because remember, if you're lipid soluble, you can enter right in through. Again, more on that later on. But some of these things do not need to be regulated by the plasma membrane. Like urea is cell waste. It's a nitrogen-based molecule. And so again, we don't want to have to go through the whole rigmarole of trying to make a special thing for urea to get out of this, outside the cell. The nice thing is it's small, nonpolar. It can diffuse right through the plasma membrane and go out into the interstitial fluid and then be absorbed by the bloodstream, and then it can move. But keep in mind that simple diffusion is all dependent, all right, on the concentration gradient. And so that concentration gradient, as long as we have more oxygen on the outside of the cell and very little of it on the inside, it's going to move from outside and in until equilibrium. So if you had 50 molecules of oxygen on the outside of the cell and 50 molecules of oxygen on the inside of the cell, oxygen is not going to move anywhere because it's equal. It's reached equilibrium. But as long as you have this concentration gradient here, a lot in one spot and a little in another, then it's going to move down the concentration gradient. So keep that in mind there. So sometimes we have to help things out, right? We have to engage in facilitated diffusion. And this, again, when we're dealing with things that aren't necessarily small and nonpolar, maybe they're small, but maybe they're charged or they are polar, like an ion. So we have to help it. So we're going to utilize some of our transport proteins that live inside the plasma membrane. So we're going to engage in facilitated diffusion, and there's two types: channel-mediated and carrier-mediated. And we'll spend a lot of time later on talking about channel-mediated. So those are the two types. And again, they're utilized for small charged or polar solids. So channel-mediated diffusion. So we have these plasma membranes that we put into the plasma, these plasma membrane proteins that are integral proteins that we put into the plasma membrane. And so they're going to allow something to move from the outside of the cell to the inside of the cell or vice versa. And so one thing that you have to know, star it, underline it, make sure you know this: channels are specific for one ion type. So that means I can't have potassium and calcium and sodium using the same channel. Doesn't happen. You got to get your own. So if sodium can only go down a sodium transport protein, potassium, the same thing. It is unique and is specific. So it's good for that. So when we're looking at channel-mediated diffusion, we have two types of channels. One, leak channels, and then we have gated channels. And so the name, we call them leak channels because they're open all the time. And when I say continuously open, they're open 24/7. They never close, like 7-Eleven, open all the time. So stuff is constantly going in and out of the cell through these leak channels, depending on what the concentration gradient is. Because remember, as long as there's a concentration gradient going on, as long as there's more stuff outside the cell or inside the cell, okay, and there's very little of it in the opposite direction, it will move. But once it's equal, things won't move. So the leak channels, they're continuously open. They're allowing stuff to flow across the cell all the time. The other one is the gated channel. And just like it says, gated channel means that there's a gate on that channel. Normally, that gate is closed until a stimuli opens it up. And when it does, it only stays open for a split second, But when it opens, then stuff can move down through the channel, depending on, right, where the concentration gradient is. And we'll talk about that. But we will really see this in detail when we talk about chapters 10 and 12, nerves and muscles. All right, so that's what we're seeing here. Here's a leak channel. They're always open. You notice lots of sodium outside the cell, not a lot inside the cell. So you can see sodium is going to go from outside to in. Vice versa. Here's a potassium leak channel. Lots of potassium outside the cell, very little of it, lots of potassium inside the cell, very little of it outside the cell. So you can see potassium is going to leave the cell. See how it kind of does that? But once we have equal amounts of sodium on both sides of the cell, it's not going to travel through the leak channel because we're at equilibrium. There's no reason. All right. The other type is called the carrier-mediated diffusion here. And look at the name, carrier. That means we're going to utilize something is literally going to carry something, move something across the plasma membrane. And that something is going to be a carrier protein. So it is going to move, right? Yes, they're small, but in this case, they're polar molecules, so they can't diffuse through the plasma membrane. We need a little bit of help here. All right, so this is what's going to happen. You're going to see this molecule come in contact with this plasma membrane protein here. It's going to change the shape of the protein, and that shape change, it will help to facilitate that movement of that molecule to the inside of the cell. So again, we're still going to be dealing with something that's going to move down the concentration gradient here, right? But it's not going to be able to make it through the plasma membrane there. So this concept is very important when we're talking about carrier-mediated diffusion, when we talk about the number of channels or carriers, right, being in the plasma membrane. If you want to increase the rate of diffusion of stuff coming into a cell, just put more carrier proteins or transport proteins in the plasma membrane, right? If we can only leave this room through that one open door, so it's going to slow us down. But if we had 5 doors on this room, it'd be a lot easier for us to walk through and to get out of here or to come in here because you don't have to wait in line. Same thing with our plasma membranes, right? If you want to be able to get more stuff from the outside of the cell, like glucose, for example, we're just going to start putting in more of these channels into the plasma membrane so we can pull in more glucose. And that's what insulin does. It makes more of these channels here so we can increase our rate of diffusion. But we can top out at that if we run out of channels there. And then if we run out of plasma membrane. So here's an example of that carrier-mediated diffusion. So there's the glucose there. It needs help coming in. So it enters in through the external portion of the channel, and you can see it changes shape. It goes through this configuration shape change here to move glucose to the inside. And so if we want to get more glucose to come into the cell, we just build more of these and just put them into the plasma membrane. Then we can get more glucose. All right, I'm going to stop here. This is a good place to stop. I know I went over a little bit. I apologize, but I needed to tell you all about this stuff.